What is testcross? Testcross is a technique to determine the genotype of an organism which shows dominant phenotype. When an organism is displaying a dominant phenotype, we do not know whether it is homozygous dominant or heterozygous. We do not know its genotype. So how can we find out? In this case, what we do is we have to cross that organism with another organism which shows recessive phenotype. Let's take an example of garden peas again. If you want to know the genotype of a garden pea plant which has purple flowers, you have to do the cross pollination, taking pollen from white flowered garden pea plants, and you have to get the garden peas and sow them. And when you sow them, if you see that all the children show only dominant phenotype, then it means this parent is homozygous dominant. Whereas when you do the cross and you sow the garden peas and you see that the children are 50% purple and 50% white flowered plants, then you say that this parent is not homozygous but heterozygous for its genotype. So remember, you have to carry out the cross pollination, taking the pollen from the recessive phenotype for that character. And when you do the cross, you sow the garden peas and you look at the children's flower color. And when you see that only of a dominant trait shows, then you conclude that this parent is homozygous dominant. Whereas if you look at children and you see that there are some white and there are purple as well, both dominant and recessive phenotype showed up, then it is called, then it is considered to be heterozygous for this character trait. So that is how testcross helps in finding out whether the dominant trait organism is homozygous for the trait or heterozygous for the trait. Let's go to the dihybrid crosses. Mendel went ahead and did some dihybrid crosses as well. So first he started off with two character plants, that means green pea plants, which would give garden pea plants which would give peas which are yellow and round in color, and other plants give green and wrinkled in shape. So he took two characters. One is a shape of the garden pea and another one is color of the garden pea. So the shape yellow, the shape round with yellow color are always in combination, whereas green color garden peas, they have wrinkled coat like this. So he called it wrinkled. Round is nothing but its seed coat is smooth. It does not have wrinkles. So he saw that these characters always go together. Garden peas, when they're yellow, the seed coat is nice and smooth. When garden peas are green in color, the seed coat is wrinkled. So he did the cross, that is cross pollination, getting pollen from one plant and putting it on another, and he saw that the green peas or the garden peas that are formed, they are yellow and round. That means green and wrinkle did not appear. This helped him to conclude that yellow color is dominant over green color, round shape is dominant over wrinkle shape. And when he sowed these seeds and he got first generation plants, and he allowed them to self-pollinate, the flowers form and they self-pollinate, and they again form the garden peas, he looked at the garden peas from the plant. And he saw that 9 out of 16 were yellow and round, 3 out of 16 were green and round, 3 out of 16 were yellow and wrinkled, and only 1 out of 16 was 1 out of 16 was green wrinkled, like one of the parent. This is a mistake here in the PowerPoint. It should have been 1 out of 16. So he said that a round shape does not always go with yellow color. It can also go with wrinkle shape or vice versa. So these characters separate, they're independent of each other. And observing this, a dihybrid cross, he said that the alleles separate from each other. Alleles for one character and another character, though they appear very commonly together, they are not linked to each other. They can separate and they can be interchanged. So this he framed and stated as law of independent assortment, in which he mentioned that each pair of alleles segregate independently of each other pairs of alleles during gamete formation. That means big Y and little y, they separate from each other, and big R, little r separate from each other. So the combination could be big Y, big R, big Y with little r, little y with big R, and little y with little r. This is just like your FOIL, first, outer, inner, last in algebra. So this is a two character cross. Remember, there are two characters, that is why you should have two letters for each character. Yellow color, because it is dominant, and this is a true breed that he started off with, so it is big Y, big Y. Round shape, again true breed, so big R, big R. Same thing, little y for green color, little r for wrinkle shape. When the alleles separate, big Y separate and big R separate, then one allele from each character is passed on to a gamete. So one big Y and one big R is given through the gamete, one little y and one little r are given through the gamete. When the gametes fertilize, they form a zygote. Zygote will undergo many cell divisions, will form an embryo, which is nothing but seed. So the seeds would have a big Y and a little y, a big R and a little r. But it will be yellow in color and round in shape because they are the dominant traits. Though they are making green color and they are supposed to be wrinkled, they appear round in shape because of the dominant trait. That's the property. When the children, which are big Y, big R, little r, they're allowed to cross with each other. Remember, when garden peas are formed in a pea pod, there will be anywhere between 4 to 6 garden peas, right? So you sow them and you get multiple plants, and these plants, you allow them to self-pollinate, then they make more garden peas. So there will be lots of grandchildren, and he observed in grandchildren how the color and the shape were going to show up. And he saw that, he observed that yellow and round did appear more in number, and green and wrinkled also came back, though it disappeared in first generation. But he also saw that yellow can be wrinkled and green can be round for the first time. So this is how you do the dihybrid cross. Guys, remember, this is the dihybrid cross. When children, hybrids, that means heterozygotes, are crossed, this is dihybrid cross. This is a two character cross. This is not a dihybrid cross. Dihybrid in true terms means when the two parents, which have two characters and they are genotypically heterozygous for each one, that is big Y, little y, big R, little r, is crossed with another big Y, little y, big R, little r. So the gametes from one parent will be big Y, big R, big Y, little r, little y, big R, little y, little r. And gametes from another parent would be big Y, big R, little y, big R, big Y, little r, little r, and you have to just mix them and write for grandchildren. Remember, always organisms have 4 letters for 2 characters, whereas gametes would have only 2 letters for each character. So that is why this is a dihybrid cross. And none of them repeat. You see that? They're all unique gametes. So this is how you make dihybrid cross and you get the count. What I ask you to do in order to find out the number of each combination is just put a little tick for yellow round, yellow round, yellow round on the side. Don't cross it off, because in case you need to redo all the work, you won't have it. If you have to answer other questions, then you won't have them. So for yellow rounds, just put a tick and then count them. For yellow round, they're all yellow round, so just put a tick, and there are 9 of them. That's how you write. And for this genotype, it should be yellow wrinkled, so put a dot. Yellow wrinkled, yellow wrinkled, yellow wrinkled, so 3. And for a green round, you can put cross maybe. So a cross on the side is going to help you count 3 of them. And one and alone is green and wrinkled. So 1 green and wrinkled. Now that you have counted everything, this is yellow round. So that's why it is 9. Now that you have counted everything and all the boxes are accounted for, you know that it is the ratio is correct. So in dihybrid cross, phenotypic ratio would be always 9 to 3 to 3 to 1. 9 will be both characters dominant, 3 will be one dominant, other recessive, and other 3 will be one recessive, one dominant, and only 1 will be both recessive. So phenotypic ratio for dihybrid cross for F2 generation, that is grandchildren, will be 9 to 3 to 3 to 1. Whereas phenotypic ratio for F1 generation will be 100%. 100% dominant trait phenotypic ratio. Genotypic ratio will be 100% heterozygous. Remember, this is heterozygous for both the characters. Okay, so again, in 2-character cross, F1 generation, 100% dominant trait, that is yellow round. Genotypic ratio, 100% heterozygous. In this case, big Y, little y, big R, little r. In dihybrid cross, that is F2 generation, phenotypic ratio will be 9 to 3 to 3 to 1. All the notes are given here. This is called law of independent assortment. If you have to do the genotypic ratio for dihybrid cross, it will be much bigger. The ratio is here. I gave it in very small font on purpose. This is a very good table to understand how the ratios will be. They're going to be always the same if you start off with the same parents. If you start off with true breed for a character, one character in F1 generation, they will be 100% dominant. In case of purple and white, 100% of them were purple in F1 generation, remember? And they will be 100% heterozygous genotypic ratio. So for F1 generation, phenotype and genotype will be 100%, whether it is monohybrid or dihybrid. Always it is going to show up 100% dominant trait for phenotype and 100% heterozygous for genotype. Doesn't matter whether it is dihybrid or monohybrid. F1 means always 100%. If it is phenotype, it is 100% dominant rate. If it is genotype, it is 100% heterozygous. Coming to monohybrid F2, that is grandchildren, the phenotypic ratio will be 75% dominant and phenotypic ratio will be 75% dominant and 25% recessive, and genotypic ratio will be 25% homozygous dominant, 50% heterozygous, and 25% homozygous recessive. For dihybrid grandchildren, phenotypic ratio will be 9 to 3 to 3 to 1. 9 will be dominant looking for both the characters, color and the shape, and 3 will be if color is dominant, shape is recessive. Other 3 will be if color is recessive, shape is dominant, and only 1 out of 16 will be both recessive. You don't have to remember the genotypic ratio of a dihybrid for F2. Just remember 3 numbers. For F1, it is going to be 100% anyways. All you have to remember is for F2 monohybrid, phenotypic ratio will be 75-25, and F2 monohybrid genotypic ratio will be 25-50-25. Whereas dihybrid F2 phenotypic ratio will be 9 to 3 to 3 to 1. All you have to remember is 3 numbers: 75-25, 3 ratios: 75-25, 25-50-25, and 9 to 3 to 3 to 1. So this is Mendelian genetics. Oops, there's E missing. Mendelism, or laws of inheritance. First law is law of segregation, which says that the 2 alleles separate, that is purple and white separate from each other, and they do not blend in or mix and disappear. They are separate, so they will end up reappearing. And second law is law of independent assortment. Alleles for one character and another character, they are independent of each other and they can be mixed and matched in many combinations. Definition of allele is it is an alternative form of a gene. In case of flower color, purple flower color, one allele, and its alternative form will be, or allele for purple flower color will be white flower color. For yellow garden pea, yellow color garden pea, its allele would be green color allele for garden peas. So make sure that you practice these crosses using the Punnett squares. All you have to do is separate the letters from each other. It's that simple. Let me do this cross. If you put a Punnett square like this, gametes from this parent will be a big G, little g. Gametes from this parent will be big G, little g. So for children, you just mix up like this. And write down. So if big G is green and little g is pink, then when you cross two parents which are heterozygous, you get 75% green color organisms and 25% pink color organism. So that will be the result of the cross. So you can perform similar cross for problem number 1 and 2 to understand how it goes. Coming to different patterns of inheritance. the pattern that we saw so far with the flower color, seed color, seed shape in garden peas is called complete dominance. That means there are two alleles in an organism, then only the dominant color will be showing and the other color will not be showing. This is called complete dominance. But it is not true in case of humans and other organisms, and even in plants many characters are there and they do not follow this complete dominance. Other patterns of inheritance or deviations to this complete dominance pattern are codominance, which is seen in AB blood group. A person with AB blood group is not showing complete dominance, but it is actually codominance. Incomplete dominance means there will be two alleles, but they together shows third trait. Pleiotropy is one gene, many effects. Example is sickle cell disease. Epistasis is one gene, affects another gene. You have to remember the Labrador dogs. For multiple alleles, remember there are many alleles in a population. There are more than two alleles in population. But when it comes to a person would have only two alleles. If a person has a big A, then the blood type of that person will be A. If a person has big A, big O, then the person will have again A blood type only, because O is recessive to A. Similarly, BB, if the genotype, then the blood type is B. Again, BO is the blood type, is a genotype, then blood type will be B again. So for a person to have O blood group, that person should be missing A and also missing B. So only when the alleles are big O, then the person would have blood group O. You know that blood group O is quite common. Coming to polygenic inheritance. What is polygenic inheritance? When a person has many alleles, then it is called polygenic inheritance. Remember, multiple and polygenic both mean many, but multiple alleles means in a population there are many alleles, but in a person there are only two. Whereas in polygenic inheritance, a person has multiple alleles. A person has more than two alleles for that character. Skin color is not due to two alleles, but it is due to six alleles, three given by one parent and three given by another parent. Then it is called polygenic inheritance. Coming to multifactorial characters. Whenever we go to beach and tan or sit, sit under sun for a long time, our skin color changes. Though skin color is due to genes, the color has changed by going to beach and sitting under the sun. It's not because our genes have changed because we were exposed to sun, but it is due to the effect of sun. The way the genes have been working changes. That is why we tan. Our skin shade changes. So it is not purely genetic. It is also affected by environment. Then it is called multifactorial character. So codominance, the many examples. The best example to remember will be AB blood type. If mother gives A, father gives B, then the child will be AB blood group. The person cannot be C blood group. It has to be both A and B. AB is a blood group. Coming to incomplete dominance, the best example would be snapdragons. When you cross snapdragon flowers, when you carry out cross-pollination between snapdragon flower plants which have red and white, you cross them, what you get in children will be pink. Red and white do not come up at all. They all will be pink whatsoever. Whereas when they make children and you sow those seeds and you get plants, you see that some plants are giving red only, other plants are giving white only, and there will be a group of plants, bigger number of group of plants, which are giving pink again. So this is called incomplete dominance. That means two traits will mix up and the third trait will appear. So intermediate, incomplete has in it, intermediate word has in it. So somewhere in between red and white, pink appears. So this is incomplete dominance. Don't think that just because dominant trait requires only one allele, all the traits that are found in population are all dominant. No, it's not true. The best example is polydactyly. Polydactyly, that means having extra fingers, is actually a dominant trait, but look around, not many people have more than 10 fingers, right? And it's a recessive trait. So recessive trait can be common as well. It depends on natural selection. Coming to multiple alleles, remember again blood groups, blood type. Blood type is either A or B or AB or O. So in population there are many alleles, and hence this is an example of multiple alleles. When you have AA, then your blood type is A. When you have blood group is A. When you have A, big A, and a little I, or you can consider this as O, then you have A again, so on and so forth. Coming to pleiotropy, one gene, many effects. Example is sickle cell disease. Remember, people in sickle cell disease, what they have is one gene for hemoglobin which is mutated. Only one gene is mutated, but because of sickle cell anemia that is given by mutated hemoglobin gene, people who have sickle cell anemia, when they go to places where there is less oxygen, such as high altitude places, or when they are too tired and exhausted physically, then they start to experience weakness and dizziness and anemia, and they start to faint, their organs start to damage, and if it is not taken care of early on, then it leads to paralysis, failure of organs, and so on and so forth. So one gene but many effects. This is called pleiotropy. You also have to practice trihybrid crosses. In this case, these are the three characters. Three characters are given. Let's say this is hair color, this is eye color, this is nose shape. Then the gametes from this parent will be big X, big Y, big Z. That is one kind. And big X. My X's are not coming out really good. Big X, big Y, and little Z. I like to write curvy little Z like this. And I think two more gametes are possible. Little X, big Y, big Z, little X, big Y, little Z. Couple of suggestions. Make sure that you always write nice curvy letters for the lowercase letters, and also know the formula how to calculate the number of unique gametes in a parent. So if this is a parent in which you're considering 3 characters at a time, um, mm, know that there will be 6 letters, 2 letters for 1 character. So the gamete will have only 3 letters. If you're considering 3 characters, 3 letters in the gamete. How will I know how many unique gametes can I get from this genotype? The formula is 2 power n. So what is n? N is how many characters are heterozygous. You can see that big X little x is heterozygous, big Z little z is heterozygous. So out of 3 characters, only 2 characters appear heterozygous. So n is how many are heterozygous. So 2 power 2. 2 power 2 will be 2 times 2. Then it will be 4. So there will be total of 4 gametes as I've shown earlier. Okay, so 2 suggestions: use the formula 2 power n. N stands for how many are heterozygous, and also use curvy letters and letters with tails for representing the recessive allele. For example, S is as big as or little s. It's confusing, right? So what I do is I make it like ampersand for little s. So use maybe a cursive writing a little bit. Somehow change the letter, clearly write what is lowercase. This is big C or little c. It's confusing, right? So what I do is I put a little tail here to represent that this is a little c. So just using the cursive letter format. Coming to epistasis. One gene will affect another gene. Remember Labrador dog coat color. Labrador dogs, they have 2 alleles for its coat color. One is black and the other one is brown. And this dog should have been actually black, because big B, dominant allele, should give black color, but it is not black. Same thing, this one should have been brown because it has little b, but it is not brown, it is gold or yellow in color. So where did this gold color come from? The reason why they're gold, they're not black or brown, is because they do not have pigment deposition G, which is big E. If this gene is not there, then they are not able to make a protein which will deposit that black pigment or the brown pigment made by another gene, made by the cells by using another gene, and hence their coat color is not black or brown, but it is yellow. So these dogs are definitely making black pigment, but because they are lacking that big E gene, they are not able to deposit that black pigment in their hair, so they are not able to appear black in color, so they appear gold color. Same deal with this brown color allele. They're not able to deposit that pigment. So this is called epistasis. One gene will affect another gene. A pigment deposition gene will affect the coat color of the dog. Coming to polygenic inheritance, skin color in population, there are many shades, but in each person it is due to 6 alleles. If we have all light color alleles given by both the parents, then the person is lightest in color. If you have combinations, there are more of dark and more or less of light, then you are on the darker side. And if you have more of light and less of dark alleles, then you're on the lighter side. So one person has many alleles for a character, not 2 alleles, but 3 and 3, 6 alleles, then it is called polygenic inheritance. When you take hydrangea plants and change the fertilizer from basic fertilizer to acidic fertilizer, it will change its color. In one type of fertilizer, it will show purple color flowers. The same plant, just because you have changed the fertilizer, it will start giving pink color flowers. This is called multifactorial character. Same thing with our skin color. When we go in sun, we are of one shade, but we will be a couple of shades darker depending on how long we have tanned ourselves under the sun, how much we have exposed ourselves to sun. Pedigrees. Pedigrees are like flow charts in which you use square and circle, square for male, circle for female. Dark color, square for the male who has the trait that you're considering, and female has a dark color circle. When you have a line between square and circle, that means they are the parents, and all the siblings are connected by one line, like branches like this. So what are pedigrees? Pedigree, as I said, is a flow chart, but it answers questions. It's not just a flow chart. It will answer a question: is this specific trait, so and so, whatever it is it dominant trait or is it recessive trait? It will just simply answer your question. So let's take the example of widow's peak. You want to know whether widow's peak is a dominant trait or recessive trait. You cannot see how the genes work, but then you can perform this on your own family, on any character that you like to know about. This kind of hairline is called widow's peak, and round hairline is called just no widow's peak. So you want to know whether widow's peak is dominant or not. All you have to do is observe the trait, whether you have it or you do not have it, and your parents have it or not, and parents, sisters and brothers have it or not, and parents, that means your grandparents, whether they have the trait or do not have the trait. So make a flow chart like this, and then simply start filling up. You say that you have a widow's peak, then you say you have big W or big W little w. You do not know. And let's say you have a sister and your sister does not have widow's peak, then you just put a circle and do not fill it. And I'm assuming that you're in case you're a girl, this is what your flow chart would look like. If you're a boy, it should have been a square. Okay, so coming to parents, let's say both the parents have widow's peak. Then you have to put big W, little w, big W, little w, because your sister does not have it. How can she not have it? She does not have it because both your parents gave little w, little w. That's why your parents have to be this. So like that, keep filling up the pedigree chart, and then you will know whether the trait is dominant or not. If it was the other way, you saw that widow's peak is not a dominant trait, it is a recessive trait. And when you put little w here for yourself, then sister will be big W, we do not know what. Then your parents have to be little w, little w. How can they have a child with one big W when they do not have big W to give, right? So our question is widow's peak a recessive trait would be wrong. So your flow chart will tell you whether the trait is dominant trait or recessive trait. Like that, you can do another example for earlobe. You can perform many tests in order to find out if there are any genetic disorders in the baby that is not born. Usually they do newborn screening, that is ultrasound, and look at fetoscopy and find out if there are any anatomical differences, whether the hands and legs have been forming okay or not. But in case you want to find out the genetic defects, then there are two ways. So the fetal testing of the chromosomes are carried out by taking out the fetus cells, that means baby which is not born yet. And one is called amniocentesis, and the other one is chorionic villus sampling, in short CVS, like your pharmacy CVS, but CVS stands for chorionic villus sampling here. So there are two ways to test fetal cells. First one is on amniocentesis, which is done 14th to 16th week of pregnancy. What the doctor does is, by looking at the baby through the ultrasound, through the scan, the doctor goes with needle through the mother's womb. They can see where the needle is going, and they get the cells around the baby in the amniotic fluid. So this is called amniocentesis, and they take the cells, they provide more food for the cells will grow, divide more in the culture plates. They're cultured, that means they are allowed to divide more outside mother, and then their DNA is extracted. The chromosomes are looked for, and if they see that the number of chromosomes are not in pairs, then they can say that there is some kind of chromosomal aberration or defect in the baby, and they can let the parents know, and parents can take the decision. But remember, this is done a little later, 14th to 16th week. It's better to do chorionic villus sampling, which is done 8th to 10th week of pregnancy. In this case, what they do is they go with a little suction tube through the birth canal, and they go to the placenta of the mother, where the baby cells are there, and they get the cells out, and they get lots of cells in this method. And they carry out different tests, biochemical tests or karyotyping, and they can find out if there is any genetic defect or chromosomal defect, and they can let the parents know whatever the results come out to be. So that is chapter 14. There are good review slides here. Please make use of them. And some pedigrees and genetics problems already solved for you.